Hello everyone and welcome back. This presentation is on double fertilization and embryogenesis. And the learning goals are the following. 1. Determine the role of stigma and synergids in pollen tube growth. 2. Explain double fertilization process in Angiosperms. 3. Identify the post-fertilization changes in the zygote and PEC. 4. Distinguish the three types of endosperm formation. 5. Depict the stages of embryo development in dicot and monocot plants. And 6. Draw a labeled diagram of a mature dicot and monocot seed. Now, the transfer of the pollen grains from the stigma cannot guarantee successful fertilization. Pollen grains face a series of problems in accomplishing their goal of delivering the male gametes to the ovule for fertilization. And all of these complex events, starting from the time of pollen deposition on the stigma to the entry of the pollen tube into the ovule, is collectively referred to as pollen-pistil interaction. And the stigma is equipped with the mechanisms to accept or reject the pollens which are deposited on its surface. Now, studies had reported many complex genetic and chemical communications or signaling between the pistil and the pollen grains. Pollens which are of the right type or the compatible pollens are accepted by the stigma, and the stigma then secretes various chemicals such as sugars, proteins, lipids or enzymes which attracts the pollen tube and thereby promotes pollen germination. On the other hand, pollens which are of the other species or which are the self-incompatible types are rejected by the stigma and prevented from pollen germination. Compatible pollens are hydrated initially on the stigma, and the vegetative cell in the pollen grain produces the pollen tube through one of its germ pores. The pollen tube then penetrates through the stigmatic tissues, and they are then further guided into the style and the ovule by the different chemical signals. You can see the emergence of the pollen tube in a germinating pollen tube in these two images. So we will be studying in vitro pollen germination as a part of our practical work once we get back to the school. Now, in the meantime, I have shared the link on an online lab in the description, and I hope this might help you to get you an idea and get yourself prepared for the lab. Anyways, coming back to the pollen grains. Now, as we all know, most of the pollens are shed at the two-cell stage. And such pollens are going to be producing the two male gametes in the pollen tube. Now, the two male gametes are produced by a single mitotic division of the generative cell. And some of the plant species, pollens can also be shed at the three-cell stage. And in these pollens, the two male gametes are already produced in the anther or before their pollination. Now, the question is, how does the pollen tube enter into the embryo sac? So, generally, most of the pollen tubes gains an easy entry through the micropyle. The two synergids which are located at the micropylar end plays a leading role in the guidance and the release of the sperm cells. And these synergids are able to produce proteins which are called as lures, as seen here in the figure. And these layers are the chemicals which can attract the pollen tube, and hence they are called as pollen tube attractant. So once the pollen tube gets into the pollen embryo sac through one of the synergids, the pollen tube along with the receptive synergids bursts open in order to release the two male gametes into the embryo sac. And the two male gametes are then to take part in double fertilization. After the successful fertilization, entry of the additional pollen tubes are blocked by the embryo sac. And this mechanism is initiated by the second synergid or the other synergid. So this second synergid cell would fuse with the fertilized central cell, and it would be thus causing the dilution of the pollen tube attractant, and at the same time there is subsequent disintegration of the second synergid, and these mechanisms are collectively going to be resulting in the prevention of polytuby, or the prevention of multiple entry of pollen tubes. And such mechanism is very similar to the response of our egg cell to the polyspermy. And there are structures which are also known as Filiform apparatus in the synergids, which also helps in the guidance of the pollen tubes entry. All right, let us now look at the process of double fertilization in angiosperms. So as we all know, the fertilization here is very unique since it involves two fusions at a time. So out of the two male gametes, one male gamete fuses with the egg cell or the female gamete, and this type of a typical fertilization is called as SYNGAMY. The other male gamete fuses with the two polar nuclei, and this type of the fusion is called as triple fusion. Now this is because it results in the fusion of three nuclei. Now SYNGAMY would give rise to a diploid zygote from the fusion of two haploid gametes, whereas triple fusion would produce a triploid cell, which is known as PEC, which stands for primary endosperm cell. So the formation of the triploid PEC is because of the fusion of three of the nuclei, haploid nuclei. So after such successful double fertilization, now the floral structures are going to be undergoing successive modifications, which are described as post-fertilization events or modifications. Some of the major modifications in the floral structures are shown in the table here. So after fertilization, ovary is going to be maturing and differentiating into the fruit, and the mature ovule is going to be becoming into the seed. While the other structures such as the two integument layers forms the two layers of the seed coat, where the outer integument would give rise to the outer seed coat called as testa, and the inner integument is going to be giving rise to the inner seed coat called as tegmen. The funicle, which is the stalk of the ovule, becomes modified into the stalk of the seed, and in some of the seeds we may also find persistent leftover nucellar tissues, and such persistent nucellus are called as perisperm. Most importantly, the diploid zygote is going to be growing and developing into a diploid embryo, and the triploid PEC is going to be maturing into a triploid endosperm. And we are going to be taking up the development of the embryo as well as the development of endosperm in this section. So let's look at the events which results in the development of endosperm in the seeds. So we need to know that the development of the endosperm precedes the development of the embryo. Endosperm tissues stocks up food reserves inside and thereby serves as a nutritive tissue for the developing embryo. Now, depending on the formation of the endosperm, it is divided into three types. The most common type of the endosperm formation is the free nuclear endosperm. Now, as shown here, in the free nuclear endosperm, the PEC cell undergoes continuous nuclear divisions without cytokinesis and cell wall formation. And consequently, it leads to a multi-nucleated condition where all of the nuclei are going to be pushed towards the periphery by the large vacuole. However, we do find incomplete or complete cell wall formation in such endosperms in the later stages. The second type of endosperm formation is called as cellular endosperm. Now, in this endosperm, every nuclear division is followed by a cytokinesis, and thereby it gives rise to a cellular mass. Now, considering the development of endosperm in the coconut, so in the coconut, primary endosperm cell has two stages of endosperm development. In the initial first stage, the primary endosperm cells are going to be undergoing successive nuclear divisions, giving rise to a free nuclear endosperm, which we commonly know as the coconut water. And this is the liquid endosperm. Now, on maturation, there is the second type of endosperm formation in the coconut. So, there is incomplete cellularization, or we can say there is the formation of the cells which is going to be forming a solid endosperm called as the white kernel, and such type of the endosperms are called as cellular endosperm. So, in the coconut, we do find two types of endosperm, the liquid endosperm, which is the coconut water, and the solid endosperm, which is the white edible kernel. Now, the third type of endosperm formation is the halobial endosperm. In this type of endosperm formation, the first or initial few divisions of the PEC are the cellular type, but the subsequent divisions are going to be all nuclear type, and hence is considered to be an intermediate type between the cellular and the free nuclear type of endosperm. So, in general, most of the endosperm tissues are used up for the embryo development. And if there is complete consumption of the endosperm, or if there is no endosperm in the mature seed, such seeds are referred to as ex-albuminous or non-endospermous seeds. Contrastingly, in some of the mature seeds, we do find persistent leftover endosperm. And such seeds which has persistent endosperms are called as albuminous or endospermous seeds. And as we have discussed earlier, so the best example, one of the best examples for the albuminous seeds or the endospermous seed is the coconut, whereas seeds such as mango seeds, bean seeds, pea seeds, or the sunflower seeds are examples for non-endospermous seeds. Next, let's try to study the process of embryogenesis, which is actually the process of formation of a multicellular embryo from a zygote. Now, let me first discuss the stages of the embryo development in dicot plants. So, in the dicot plants, the first stage is called as the two-celled stage. Now, during this stage, zygote divides transversely, resulting in the formation of two unequal cells, which are the smaller terminal cell towards the chalaza and a bigger basal cell towards the micropyle. Now, the second stage in the embryo development is called as proembryo stage. And during this stage, the basal cell divides further and it's going to be contributing to the formation of a long chain of cells called as suspensor cells. And these suspensors are helpful in pushing the embryo down into the endosperm. Whereas the terminal cell divides further to give rise to the proembryo. So, it's actually the terminal cell which gives rise to the embryo. And if a terminal cell gives rise to a four-celled proembryo, then it's called as a quadrant proembryo, whereas a terminal cell which has divided and formed an eight-celled proembryo, such a proembryo is called as an octant proembryo. Out of the 8 cells of the octant proembryo, the 4 cells which are near to the suspensor are going to be giving rise to the hypocotyl. So, hypocotyl are the embryonic parts which lies below the cotyledon. And the other 4 cells which are towards the chalazal end are going to be giving rise to the cotyledon and the plumule. Cotyledon is actually an embryonic leaf which stores food in the developing seed, while the plumule is the shoot meristem, which is then going to be giving rise to the shoot system. So, after this proembryo stage, the next stage is called as the globular stage. Now, this stage is formed by the division of the proembryo in different planes, and as a result of which we have the cells arranged in many layers. The outer layers of the cells are going to be dividing and differentiating into epidermis, whereas the inner cells are going to be differentiating into the ground tissues such as the cortex, the vascular tissues, and the pith. Now, with the continuous growth, the embryo then enters into the next stage, which is called as the heart-shaped and torpedo-shaped stage. So, in this stage, the embryo has formed the 2 cotyledons along with the shoot apical meristem, hypocotyl and radicle. And all these structures, including the 2 cotyledons, are going to be organized in such a way that it gives a kind of a heart shape or a torpedo shape to the embryo. And finally, the last stage is the mature embryo stage. Now, in the mature embryo stage, the cotyledons have grown sufficiently long enough that they are going to be showing a bend in order to be fitting themselves inside the seed. And by this stage, the suspensors are all going to be degenerated. And the embryonal axis are going to be consisting of 2 meristems, embryonic meristems at their ends. So, the shoot meristems, which are called as plumule, are present above the cotyledon, whereas the root meristems, which are called as the radicle, lies below the cotyledon. And the region which is between the plumule and the point of attachment of the cotyledon is called as epicotyl, and the region which is between the radicle and the point of attachment of the cotyledon is called as hypocotyl. So, actually epicotyl lies above the cotyledon and hypocotyl lies below the cotyledon. So, in this way we understand that the embryo development in dicot plants includes stages such as the two-cell stage, proembryo stage, globular stage, the heart and torpedo shaped stage, and finally the mature stage. Now, in the monocots, the stages of embryogenesis includes proembryo stage, globular stage, scutellar stage, and coleoptilar stage. The basic developmental pattern remains very similar to that of the dicot plants. However, the basal cell doesn't divide any further, and it gets modified into a large vesicular cell. So, in the monocots, basal cells are not responsible for the formation of the suspensors, whereas suspensors are formed by the division of the terminal cells. And in the scutellar stage, the monocot embryo develops a thin, papery single cotyledon, which is technically referred to as scutellum, and hence the name to the stage. So, the embryonic meristems, which are the plumule and radicle, has specialized sheath surrounding its tips. So, the sheath which is present on the plumule is called as coleoptile, and the sheath which is present on the radicle is called as coleorhiza. And such an embryonic stage where we have the formation of the coleoptile and coleorhiza is referred to as the coleoptilar stage. The thin cotyledon, which are known as the scutellum, aren't responsible for storing food as in the cases of dicot plants, but these are important in terms of acting as a conduit, or it helps in the conduction of the nutrients from the endosperm to the embryo. So, in this way we find that the stages of embryo development in the monocot includes the 4 stages, which are the proembryo stage, globular stage, scutellar stage, and the coleoptilar stage. So, the main differences are that the monocots have a single cotyledon known as scutellum, whereas the dicots have 2 cotyledons. The cotyledons are not involved in storage of food in the monocots, whereas in dicots the cotyledons store food in the cotyledons. And the shoot meristems and the root meristems are protected by the protective sheath known as coleoptile and coleorhiza in the monocots, which are absent in the dicot seed. So, with this I come to the end of today's presentation on double fertilization and embryogenesis, and I hope I have made sense to you. Now, before I sign out, I want to tell that you all need to check Google Classroom for the assignments and other updates.